Okay, so good afternoon, everybody. It's 1 o'clock according to my clock, so I'm going to go ahead and get started. I had a nice weekend. Excuse me. One quick announcement. I just got an email from Dr. Tang, the 201 lead, and it has been decided that all summer classes at Front Range are going to be online as well. I'm not sure if everybody online classes or if anyone has seen that same announcement, but for those of you who are enrolled in summer classes, I just want to make sure that you are all aware that it looks like summer classes are going to be online as well. So just thought I'd share that information with everybody. So that being said, today's agenda, we're going to focus primarily on digestive system physiology. We covered digestive system anatomy and histology when we met last week. That way, if we focus almost exclusively on digestive system physiology today, we'll easily be able to get through the material, which will give us all of Wednesday to review for next Monday's exam, meaning next Wednesday, we'll review respiratory system, digestive system, and I'll also go over exam format then. I haven't put the exam together yet. I'll be doing that tomorrow. So I don't really address format today anyways. So that being said, again, focusing on digestive system physiology today, let's just kind of open the floor, and I'm here to answer any questions that you may have. What I'm really hoping to cover today is when it comes to digestive system physiology, what I want to know is understand the function of the main digestive system products that digest macronutrients, enzymes, acid, and bile. Know where they're produced, know what macromolecule each digests, fats, proteins, carbohydrates, and know where in the digestive tract that digestion occurs. And then also definitely make sure that you understand the anatomy and the physiology of the liver. Understand what type of blood flows into the liver, what type of enzymatic processing occurs in the liver, and understand how blood flows out of the liver. So that being said, I'm just curious if someone can speak up to this. When I have an icon on my screen that shows, when I click it, it's the participants icon. It looks like a silhouette of a person. When I click on it, it gives me a list of everybody who's attending. Can you all see that same list?
I can see it.
Okay, great. Then I don't need to announce who all is here. And I appreciate the fact that a few of you have sent hello messages. So Elizabeth, what sucks? You typed.
Oh, I mentioned that in regards to.
I'm sorry, I didn't hear what you just said.
I said I mentioned that in regards to the online classes announcement.
Okay, I understand. And also, I appreciate Rachel typing in a comment asking people to. So again, remember, unless you're actively speaking, asking a question, commenting, or answering a question, please mute your audio. I just discovered this morning that I can mute you. I can mute anybody on my end. So I'll start doing that as well. But again, yeah, Rachel, I appreciate you typing a comment asking others to mute their audio. I can start doing that as well. I had an issue with my morning class. A student, I think she was at work. She left her computer screen and her background was so noisy that no one could hear each other. And that's when I realized that, no, I could have muted her the whole time. Okay, so that being said, questions. What questions do you all have regarding digestive system physiology? These can be virtual lab questions, questions about Wendy Riggs's videos, study guide questions, PowerPoint questions. Now and Wednesday are the time to ask.
I have a question.
Okay, this is Liz.
Yeah, sorry. Um, so it's about the virtual lab, and I started doing the respiratory system, and I had a little bit of a hard time because I obviously we never really physically saw the models, and some major structures are, pretty obvious, but some little things are like, oh, not sure what you're talking about.
Okay.
Um, for example, it was a median, like half head, and there was like a little spot that it was like, what the hell are you pointing at? And what is it?
Question you're talking about. It was probably one of the questions asking you to identify the tonsils.
No, I'm not sure what it was, but it was like a bony structure. Um,
Okay. Maybe pointing
like,
thyroid bone. Um,
it's not very clear, so I had to go to YouTube and like search people talking about that model and like,
okay.
um,
So one thing about those questions, yeah, like you said, most of the questions in the virtual lab assignments, yeah, most of the structures they're pointing to are pretty obvious, but on occasion, they might be pointing to something so small and that structure is highlighted. You can't actually see the structure because the highlight over it blocks the structure. Um, of course, you with those virtual assignments, those virtual lab assignments, because they are open note, open book. Of course, you can refer to the internet or your textbook or any other source for answers.
mm,
So I just want to, I guess, emphasize one of those is basically a multiple choice question. So if you're not sure what specific structure is being pointed to, you can probably use, the process of elimination and
right.
rule out what it's not. That should help. And then also it's set up so that you can obviously take it over and over again. So if for some reason you get that one wrong, and I need to thank Clara for this, who she's not with us this afternoon. She pointed out the fact that I initially had the settings set up uh, incorrectly, or I modified the settings so that with those virtual lab assignments, as soon as you're done with the whole assignment, it will automatically show you, it'll give you a detailed report showing you every question you got wrong and why you got it wrong. So if you go back and take it again, you'll be able to answer that one correctly. But to some extent, it's just obviously I have no control over the pictures in the questions. I control which questions I included, but admittedly, some of those anatomical identification questions on the virtual lab assignment weren't perfect. But yeah, the process of elimination with the multiple choice options and looking at the detailed report once you're done should at the very least help you answer it correctly the second time.
Yeah, so my question essentially is, can we expect structures like that in the on the exam?
Yes, I am going to um, use images almost exactly like that, both anatomy images and uh, microscopic images. In fact, when I put the exam together tomorrow, I'm going to
Mm.
try to choose to pull questions from that same question bank. So you might see some of those, many of those exact same questions and images.
Okay. Okay, good to know. Oh, and then it so I had a little discrepancy. Is the exam on the 21st or the 25th? For some
Oh,
reason,
yeah. So the exam is going to be on Monday. Let me pull up my calendar. I'm pretty sure Monday is the 20th. Yes. Did I have it listed in my updated schedule as the 21st?
it was some other date I can remember. So
Okay.
I just want to clarify.
You asked. So yeah, our next exam. Whoops, didn't mean to open that. Our next exam will be a week from today, which is Monday the 20th. So for those of you in my afternoon class, obviously at 1 o'clock next Monday, our exam starts. Liz, I know that you're in my morning class, so obviously your exam will start at 9:30.
Yeah.
A reminder that by 9:30 next Monday, when the exam starts, your Connect homework assignment will be due. The Connect virtual lab assignments will be due, and you'll have to submit your lab exercise, pictures of your lab or a PDF file of your lab to me through D2L. I still haven't put together that folder in D2L. I'll have that tomorrow with the posting about it tomorrow as well. But the short answer is, yeah, our next exam is exactly 1 week from today, which is the 20th, 4/20. Easy date to remember in Colorado.
Yeah. Okay, thank you.
You're welcome. Okay, so let me ask again, does anybody have any specific questions? If not, I am going to give a brief lecture explanation on basically digestive products produced in the stomach and the specific cell type that produces the products. I'll give a quick description on the production and function of bile. We'll quickly talk about the 4 digestive enzymes produced in the pancreas. And I'll take a closer look at liver histology and really describing liver function in terms of how the liver processes newly absorbed nutrients and how the liver makes bile from blood that flows through the liver. So first of all, does anybody have any specific questions about stomach digestive physiology? Okay, so I think that is CTBAI. Is that Clara? Doesn't ultimately matter. I'm just curious. I think it is. Okay, so that being said, I'm going to switch over to my share screen and pull up some information from my PowerPoint. All right, so share screen. Close that. Okay, so let's see here. Starting with my PowerPoint slide number 23. So to gauge what time delay we have, can someone confirm whether or not you can see my enlarged slide number 23, gastric glands and pits?
We can see it.
Okay, good. In my about a 5 to 10 second delay. So I don't want to start talking about a slide until I know you all can see it. Okay, so think of it this way. When it comes to digestive, we talked about the digestive system anatomy, right? This continuous tract from the mouth through the anus. We talked about the histology, right, of that digestive tract, describing the 4 tissue layers that make up the tract and how those tissue layers differ between the esophagus and the stomach and the small intestine and the large. So today I really want to focus on the digestive products that are either produced by cells lining the digestive tract or by the accessory organs, liver, gallbladder, pancreas, that secrete their products into the digestive tract. So, excuse me. So starting with saliva, we talked about saliva a little bit last time. The salivary glands produce and secrete saliva into the mouth, into the oral cavity, primarily to moisten food, right? So that when we chew food and it mixes with saliva, it turns into a moistened ball or a bolus that can be swallowed. It's virtually impossible to swallow dry food, right? Try crumbling up a cracker and just swallowing it. You can't until that mixes with saliva. Well, There are some digestive enzymes found in saliva. Ultimately, they play such a minor role. They're almost insignificant. Yeah, almost. So what it comes down to is this. In our saliva, there's a little bit of salivary amylase. And amylase is an enzyme that enzymatically breaks down carbohydrates. Our saliva also contains a lipase, salivary lipase. This is an enzyme that starts enzymatically breaking down fat. And our saliva also contains lysozyme, an enzyme that is antibacterial. It targets and enzymatically starts breaking down bacteria. Really, really minor. These processes just begin in the saliva. Most digestion continues in the stomach, and then most chemical digestion occurs in the small intestine. So, in terms of digestive products, there's saliva, there's stomach secretions or stomach juice, pancreatic secretions or pancreatic juice, and then bile produced by the liver, stored and concentrated in the gallbladder. So let's first take a look at digestive products produced in the stomach. Now, let me scroll back one slide. So can everyone now see my slide number 22?
Yeah.
Okay. Yeah. So again, we talked about this last time. When we look at the stomach, the histology of the stomach, again, always look from the inside, the lumen, out to the muscle. So what's unique about the mucosa, the mucous membrane lining of the stomach, is that it contains thousands, maybe millions of gastric pits that lead to our gastric glands. So again, what's the difference between a gastric pit and a gastric gland? Well, the gastric pit is this opening on the surface of the epithelial lining, right? The opening into the lumen that works its way down submucosa. That typically, the point I'm trying to make is typically one gastric pit will branch off into two gastric glands. Sometimes one gastric pit leads to one gastric gland, right? Oftentimes one gastric pit will lead to two gastric glands. Well, there are specialized cells that line the gastric pits and gastric glands that produce key digestive products, right? So scrolling forward to slide number 23, what we're going to see is there are four main cell types that line and form our gastric pits and glands. Now, under a microscope, these all look the same. They don't look as distinctly different from each other as they do on this cartoonish drawing. So I wouldn't ask you to be able to identify, for example, a chief cell under the microscope. They all look the same. I want you to know the function of these different cells. So first and foremost, our stomachs, all of our GI tract, stomach through intestines, are coated with a relatively thick layer of mucus. This protects, in the case of the stomach, the entire lumen of the stomach is coated with mucus. This mucus protects the lining of the stomach from the very digestive products, hydrochloric acid, and a very potent enzyme called pepsin. Mucus coating the stomach protects the stomach lining from being digested from products produced in these gastric pits and glands. Now, mucus cells are found up near the opening of the gastric pit into the lumen. This is sometimes referred to as the neck of the gastric pit. So sometimes these mucus cells are called mucus neck cells, right? Again, neck doesn't mean your neck. It means the neck, the opening of this gastric pit. But mucus cells produce mucus that coats the lining of the stomach, protecting it from hydrochloric acid and digestive enzymes. Parietal cells. Parietal cells are these specialized cells lining our gastric pits and glands that produce hydrochloric acid. So think there's an L in parietal, there's an L in hydrochloric acid. One way to remember that hydrochloric acid is secreted by parietal cells. Now, hydrochloric acid is an incredibly strong acid. It has a pH of about 2 or less. Hydrochloric acid will dissolve anything, literally, well, almost anything. You can dissolve metal, you can dissolve nails, you can dissolve aluminum, not to mention proteins, carbohydrates, and fats. If you literally had a glass jar full of someone's stomach secretions, someone's stomach acid, you could literally put a nail in there and it would dissolve it. You know, give it half an hour to an hour and it would dissolve it. For our purposes, I want you to know that hydrochloric acid produced by parietal cells is highly effective at digesting proteins. Again, hydrochloric acid can digest anything, but again, for our purposes, the most significant digestion that occurs in the stomach is the digestion of proteins, thanks to hydrochloric acid produced by parietal cells and an enzyme called pepsin that's produced by chief cells. Now, just to make a quick note, chief cells also secrete a lipase, an enzyme that breaks down fats. Maybe 15% of all fat digestion occurs in the stomach. Most fat digestion is occurring in the small intestine thanks to a lipase from the pancreas, pancreatic lipase. And we'll talk about that next. So, mucus cells produce mucus to coat the lining of the stomach, protecting it from digestive products. Parietal cells secrete hydrochloric acid, which is a potent digester of proteins. Hydrochloric acid is also involved in the conversion of pepsinogen, which is an inactive enzyme, into its active form, pepsin. So I'm going to scroll forward 2 slides. Please let me know if you can see my slide number 25.
Yeah, we can see it.
Okay, great, thank you. So it sounds like our time delay is only is very brief. So I won't, I'm not going to ask that every time I switch slides. So one thing I really want to draw out when it comes to this slide number 25, I really like this slide, but it has one glaring error. This slide is showing how hydrochloric acid produced by parietal cells helps convert pepsinogen, which is produced by chief cells. Pepsinogen, any enzyme with the suffix ogen, that indicates that this is an inactive enzyme. Hydrochloric acid starts digesting pepsinogen, breaking off a branch of amino acids, turning it into its active form, pepsin. Well, this image makes it look like this is all happening in the submucosa. It's not. Hydrochloric acid, pepsinogen, these products get secreted into the gastric gland itself, into the lumen of the gland itself. Then those products get secreted into the lumen of the stomach. So this chemical reaction that we're seeing here, this is actually occurring in the lumen of the stomach, not in the submucosa itself. My point is just this. This is a rare example of a positive feedback, right? Most body functions, 99.99% of all body functions are regulated through classic negative feedback, right? Blood sugar gets too high, mechanisms kick in to bring it back down. Blood sugar gets too low, mechanisms kick in to bring it back up, right? Classic negative feedback. Well, I know I've mentioned throughout this semester examples of positive feedback. There's really only a few, 3 that come to mind. Childbirth is one example of a positive feedback mechanism. When an infant, right, is during childbirth, when an infant, usually head first, when an infant's head pushes through the birth canal, stretching the uterus, that stretching of the uterus sends a signal back to the brain, releasing oxytocin, causing the uterus to contract more, pushing the baby's head further down the birth canal, pushing the uterus more stretch equals more contraction, stretch equals more contraction until childbirth, right? That's an example of positive feedback. The activation of blood clotting proteins. Typically activate one protein, one blood clotting protein activates more, which in turn activates more, which activates more, which activates more. Another example of positive feedback. Pepsin both hydrochloric acid and pepsin can activate pepsinogen into more pepsin. So if I get a little bit of pepsin that's formed, thanks to hydrochloric acid, pepsin will in turn break down pepsinogen, making more pepsin. More pepsin in turn leads to the production or the activation of even more pepsin. Point is, this is a very rare example of how a positive feedback mechanism can drive a body function. In this case, the production of a highly potent protein digester. So both hydrochloric acid and pepsin can digest proteins. Both hydrochloric acid and pepsin can convert inactive pepsinogen into highly active pepsin. The take-home message is this. In the stomach, the most chemical digestion is the digestion of proteins, thanks to hydrochloric acid from parietal cells and pepsinogen, which gets turned into pepsin from chief cells. So now I'm going to scroll back to slide number 23. We can see that there's one other specialized cell to talk about, found in our gastric glands. At the very bottom or base of the gland, there are cells that are just generically called G cells. They're called G cells because they produce a hormone gastrin. We talked about gastrin briefly at the beginning of the semester, right? Gastric refers to stomach. Gastrin is a hormone. It doesn't get secreted into the lumen of the gland and pit, and then ultimately into the lumen of the stomach. Being a hormone, gastrin gets secreted into the blood vessels that are found in the submucosa of the stomach. Gastrin enters the bloodstream, travels throughout all the blood vessels of the stomach, and it directly activates parietal cells to make hydrochloric acid, chief cells to make pepsinogen. So really what it comes down to is the biggest, strongest stimulus for stomach activity is the filling and stretching of the stomach. When you swallow a bolus of food, travels down the pharynx, esophagus into the stomach. When that bolus enters the stomach, it'll fill and expand the stomach. Stretching of the stomach activates the secretion of hydrochloric acid, the secretion of pepsinogen, and the secretion of gastrin. Gastrin in turn causes more hydrochloric acid to get produced by parietal cells, more pepsinogen to get produced by chief cells. The gastrin also stimulates the 3 layers of muscle surrounding the stomach to start contracting, ensuring that the food that you just swallowed is not only, as it's mixing with hydrochloric acid and pepsin, the contracting causes it to mix, and that relatively vigorous mixing is highly effective at beginning the breakdown of proteins. So again, I know that most of you probably thought the stomach was the location of most chemical digestion. Your stomach is almost more of a holding tank. Some protein digestion occurs there. I can't emphasize this enough. Most chemical digestion and virtually all nutrient absorption is occurring in the small intestine. The stomach just kind of gets the process started. Again, it's hydrochloric acid from parietal cells, pepsin from chief cells, and gastrin, right, from G cells. Okay, so that being said, and as I mentioned before, almost everything that I'm describing now is in text format. In addition to pepsinogen, chief cells also produce a lipase, right? Gastric lipase, an enzyme that can break down fats. As I mentioned before, only a small amount of fat digestion, the enzymatic breakdown of fats, is occurring in the stomach. The vast majority of that, just like the vast majority of carbohydrate digestion and the vast majority of protein digestion, is occurring in the small intestine. More specifically, the first foot of the small intestine, right? That area known as the duodenum. Okay, so that being said, again, I'm going through this pretty quick. Hopefully, I'm expecting everyone has already read through this material, has a decent understanding of it. We can go back through this again on Wednesday when we review. but that was a real quick run through description of the major digestive products produced by the stomach. Any specific questions on that information I just shared? Okay, so moving, sorry, jumping all over the place. Let me find the right slide. Here's the one I'm looking for. So hopefully everyone can now see my slide number 38. So again, let me know if you don't see what I'm talking about. Otherwise, I'm going to stop asking if you can see every slide that I jump to. If you can't, speak up, let me know. Hopefully everyone is now seeing my slide number 38. So what this slide is showing is the relationship between the duodenum, the beginning of the small intestine, the pancreas, and bile ducts. So right here on this image, this is essentially a cross section through or just past the pyloric sphincter. So this location here is essentially where food that is partially digested in the stomach, once it's done in the stomach, that food, aka chyme, right? Partially digested food is now chyme, chyme. Chyme enters the duodenum and it's only partially digested. It's secretions from the pancreas and bile from the liver that enter the duodenum that drive the remaining chemical digestion of fats, proteins, carbohydrates. Once chemical digestion is complete in the beginning of the small intestine, as that chyme travels through the rest of the small intestine, all of those nutrients that were digested then get absorbed in the bloodstream. So let's first talk about bile. Bile, produced in the liver, stored and concentrated in the gallbladder, ultimately secreted into the duodenum. So think of it this way. The only time, let me back that up. Bile is constantly being made in the liver. 24 hours a day, our livers produce bile. When there's also chyme in the small intestine, bile, so basically right at the top of the screen is where the liver. Bile is going to flow from the liver through a series of ducts straight into the small intestine. Okay? When the small intestine is digesting food. Well, that only occurs maybe 3 times a day. Most of the time, there is not chyme in your small intestine. Therefore, the opening from the ducts into the duodenum is closed. When there's no chyme in your small intestine, bile doesn't enter your small intestine. It actually kind of backflows up and into the gallbladder. The gallbladder stores bile, but it also concentrates it. What that means is bile is secreted. It's a pretty watery, diluted secretion. When it backflows into the gallbladder, cells lining the gallbladder remove most of the water from that bile, turning from a liquid into a pretty dense paste. If we have the opportunity to dissect the kidneys, and if we cut open the gallbladders, we would see that the gallbladders are full of a really dark green, almost greenish brown bile. It's so concentrated, it almost has the texture of toothpaste, right? It's quite concentrated. Well, bile, first of all, what is it? What does it do? Why is bile typically greenish brown? Well, basically, bile, functionally speaking, bile from the gallbladder, its main function is to emulsify fats. Now, emulsification is not the same as enzymatic degradation. Emulsification is a process that just physically shears or shreds fat molecules into smaller and smaller pieces. Enzymatic degradation involves the chemical breakdown of those molecules. So fat is emulsified by bile That's a physical process. It's like when you chomp on your food with your teeth, that is physically shredding our food. Bile functions in a similar way. It physically shreds it because bile is made of what are called bile salts. Well, what I want you to know is this. There we go. This is information that's spelled out on my slide number 35. So bile. What is bile made of? What it comes down to is this. Bile is the only way that our body can get rid of extra cholesterol is by secreting as bile. So bile is basically a combination of cholesterol and hemoglobin, excuse me, hemoglobin breakdown byproducts, right? This is all described in my slide 35. Hemoglobin, right? Found in our red blood cells. Our red blood cells only live 3 to 4 months. They get broken down and recycled. The hemoglobin within those dying, worn out red blood cells all gets broken down. A lot of it gets recycled and used to make more hemoglobin, but a lot of the hemoglobin breakdown byproducts have to get excreted out of the body as waste. Now, in the next unit, we're going to talk about the urinary system. Our kidneys play a main role in filtering waste products out of our bloodstream. Well, a lot of these hemoglobin breakdown byproducts are too big to get filtered out of our kidneys, so they get excreted out of our livers. When hemoglobin breakdown byproducts, right, like bilirubin, bile is called bile because it's mostly composed of bilirubin. Bilirubin, a breakdown byproduct of hemoglobin, combines with cholesterol, and that gets secreted out of the liver as a waste product. But this waste product has a really important function. It shreds fat into small little microscopic droplets, small enough that lipase enzymes can now enzymatically break it down into even smaller triglycerides, which can then get absorbed through the intestinal wall. So, going back to my image here, bile from the liver gets secreted in the duodenum. It physically shreds fat into smaller little molecules. So, that being said, before we talk about the pancreas, scroll back to my slide number 32. So to really understand bile production, we have to understand overall liver function. Now, what we can see here, looking at both slide 33, a microscopic image of liver tissue, and slide 32, kind of a cartoonish drawing of liver tissue. When seen under the microscope, the liver, it's one of the most easily recognizable tissues because the liver tissue is composed of and compartmentalized into these hexagon-shaped structures called lobules. Here on 33, slide number 33, this microscopic image is showing most of one complete cross-sectioned lobule. And what we see here is, functionally speaking, overall, we know that our livers are associated with the breakdown of hormones, right? One of the reasons why hormones, some have really short half-lives, well, a lot of foreign substances, drugs, for example, aspirin, penicillin, acetaminophen, right? The reason why if you take an aspirin, it's usually only effective for a couple hours, because as aspirin gets digested, absorbed in the bloodstream, it travels through the liver, and specialized cells called hepatocytes that make up our liver lobules recognize foreign substances from normal substances and enzymatically process them. So enzymatic processing typically means enzymatic degradation. Enzymatic processing means producing from cholesterol and hemoglobin breakdown byproducts. Liver processing also can mean taking extra glucose molecules, linking them together as glycogen and storing that glycogen in the liver. So what it comes down to is this. Ingestion brings food in. Digestion breaks that food down into small enough sized particles that can get absorbed into our bloodstream. This is occurring in the small intestine. All venous blood from the small intestine first travels to the liver. So all of those newly absorbed nutrients in that blood first travel to the liver so the liver can process this blood. Well, enzymatic processing in the liver also involves the conversion of inactive vitamins into active vitamins, or the conversion of provitamins into active vitamins. Oftentimes, the vitamins found in the food that we eat are absorbed in an inactive state. Those vitamins travel into the liver where cells, these hepatocytes in the liver, enzymatically convert provitamins to active vitamins. They enzymatically break down any foreign substances like aspirin, penicillin, other drugs. They help enzymatically break down alcohol, right? We all should know that our livers play a major role in breaking down alcohol that we consume. If there's extra sugar in the food that we eat, there's going to be more sugar absorbed into our bloodstream, more sugar enters the liver. Our livers will store that extra sugar. All examples of enzymatic processing. So what it comes down to is this. Notice that at the corner, each corner of these liver lobules, we see 3 distinct vessels. Each group of 3 vessels forms what's called a triad. Now, these vessels, one blue, one red, one green, 2 of these carry blood in, one carries bile out. So starting with blue, this blue vessel, right, or these blue vessels I see at each corner of the hepatic lobule, these are branches off the hepatic portal vein. The hepatic portal vein. That's the vein that's bringing blood from the intestines to the liver. What's in that blood? All of those newly absorbed nutrients. There might be some absorbed drugs if you just swallowed an aspirin. There also could potentially be harmful substances like alcohol, right? If you just consumed alcohol. All that blood first flows to the liver via the hepatic portal vein, and branches of that hepatic portal vein travel to all of these corners of the lobules. We also see that the liver receives oxygenated blood through the hepatic artery, and in that oxygenated blood, obviously, is oxygen to fuel these hepatocytes, right? Fuel them for their enzymatic processing. Well, these cells also need glucose. The glucose that we previously consumed that's flowing through our bloodstream, it travels into the liver to fuel these hepatocytes. So what it comes down to is this. Notice if we look at one lobule here in the cartoonist drawing, scrolling ahead to slide 33 on my microscopic image, here we see a textbook triad, a branch of the hepatic portal vein, a branch of the hepatic artery, bringing blood in, and a bile duct tool bringing bile out. So as this blood enters the liver via the hepatic portal vein, that branches, and that blood then flows through these little channels from the outside of the lobule into the center, emptying into the central vein. These channels that run from the corners of the lobule to the inside or the center, these are called sinusoids. Sinusoids are these specialized capillaries that carry blood from both the hepatic portal vein branch and the hepatic artery. We can see from this diagram right here that once these blood vessels enter the lobule, they converge so that blood flowing through the sinusoid is blood that just came from the intestines with all these newly absorbed nutrients, but also oxygenated blood that has been flowing right through our systemic circulation. As that mixed blood flows through the sinusoids down towards the central vein, these big cube-shaped hepatocytes that line the sinusoids are removing alcohol, removing drugs, enzymatically processing vitamins, maybe absorbing glucose and storing it as glycogen, okay? Absorbing hormones and enzymatically breaking those down, right? That's what we mean by enzymatic processing. So that blood that enters the sinusoid, travels down the sinusoid, eventually emptying into the central vein. Once that blood enters the central vein, it's done being processed. The central veins all converge into right and left hepatic veins. The hepatic veins then empty that blood into the inferior vena cava, which carries it to the heart, and then the heart can now pump all of these processed nutrients and vitamins out through the systemic circulation. Going the wrong way. We take a look at slide 34. This is an grossly overly simplistic representation of what I just mentioned. This is showing blood from the intestines and all these newly absorbed nutrients entering the liver, being flowed into the lobules, mixing with oxygenated blood from the hepatic artery. The lobules process that blood, and when they're done processing that blood can now enter the inferior vena cava, travel to the heart, and the heart can now pump all of these newly absorbed and processed nutrients out to tissues like the brain and skeletal muscle and the heart's myocardium, the tissues that need all of these newly absorbed sugar molecules and fat molecules and protein molecules. The last thing I want to mention, well, what about bile? Well, let's say you just ate a meal really high in cholesterol. That cholesterol gets digested, absorbed in the intestines. It's going to flow into the liver via the hepatic portal vein branch. Maybe you also have a high concentration of dead and dying red blood cells and broken down hemoglobin molecules. Those are going to enter the lobule via the hepatic artery. Hemoglobin breakdown byproducts are going to mix with cholesterol, flow through the sinusoid, and as they're flowing through the sinusoids, these hepatocytes are going to remove those substances, combine them together to make bile, and then bile exits from the inside out through these bile ductules. There's a series of the smallest little channels for bile called canaliculi. Once bile is produced in the hepatocyte, it gets secreted into these bile canaliculi. Then they flow into these bile ductules, eventually out the liver and into the small intestine. From the liver to the small intestine, that's where we see these series of ducts. Now, for those of you who have completed the digestive system virtual lab, you notice that I included quite a few questions about identifying, right, these bile passages. What it comes down to is this: from the liver to the small intestine, the network of vessels that carry bile from the liver to the small intestine, it's all a series of two vessels converging into one. You see the right and left hepatic ducts are the ducts that carry bile out of the liver. As soon as they exit the liver, they converge into a common hepatic duct. Well, the duct that connects the gallbladder to this network of vessels is called the cystic duct. The two hepatic ducts, right and left, converge, forming the common hepatic duct. The common hepatic duct and the cystic duct converge, forming the bile duct. This is sometimes called the common bile duct. The bile duct converges with the pancreatic duct, which is carrying digestive enzymes from the pancreas into the small intestine, and they both converge and open into the small intestine. So that as soon as chyme digested in the stomach enters the small intestine, it immediately mixes with bile and pancreatic secretions to complete chemical digestion, so that then those digested nutrients can be absorbed. Okay, so that was a lot of me talking. I want to next see if anybody has any specific questions regarding anything I just covered.
Ryan, this is Rolando.
Hi, Rolando.
Question is, in the stomach, when you mentioned that the stretch receptors trigger what triggers, is that a similar mechanism here in the duodenum? Do they know this one
Yes.
that triggers the release?
Question. So it's once the duodenum fills, right, with chyme, the filling and stretching of the duodenum opens up. There's a sphincter here called the hepatopancreatic sphincter. Hepato for liver, pancreatic for pancreas. When the duodenum is empty, this sphincter is closed. When the duodenum stretches, some of those stretch receptors directly form a reflex arc that directly connects to the muscles that create the sphincter, causing them to open, ensuring that the duodenum only receives bile and pancreatic secretions when it is full of chyme. And when it's empty, that sphincter stays closed, nothing, no bile, no pancreatic secretions enter the duodenum when it's not full. Did that answer your question?
Thank you.
Okay, great. Okay, does anybody have questions about bile, how it's produced, where it's produced, how it enters the small intestine, and what it does in the small intestine? Right? It emulsifies fats.
This is Liz. I don't have a question, but I have a comment. I watched this really cool video explaining the process of bile creation, and I can share it in the comments or something. But it was very helpful.
By all means, type in the link in the comment section.
Mhm.
Is it an animation or is it like an endoscopic?
It's like a lecture from another somewhere,
Okay.
but I just found it useful.
Yeah, feel free, you and anyone else, feel free to share that information. Yeah, and the best way is, yeah, type in the link into the chat section of this discussion.
Yeah, I'm looking for it right now.
Okay, great. Okay, so next, now that we talked about hydrochloric acid and pepsin production in the stomach, driving protein digestion, bile production and secretion from the liver, aiding in fat digestion through emulsification, let's next take a look at the 4 digestive enzymes produced in the pancreas. Okay, so let's start here. Hopefully everyone can now see my slide number 66, microscopic view of the pancreas. Again, this slide came from that digestive system histology PowerPoint that I added to D2L last week. So we've already looked at the pancreas pretty closely when we covered the endocrine system. Well, we only looked at part of the pancreas. Remember, we learned that the pancreas, only about 1% overall of pancreatic tissue, right, is composed of these pancreatic islets, these beach ball looking structures. It's here in the islets where we find the cells that make insulin and glucagon to regulate blood glucose levels. 99% of your pancreas is made up of these acinar cells, sometimes pronounced as cynar cells, but these acinar cells are the cells that produce digestive enzymes. Well, hormones produced by cells in the islets get secreted into the bloodstream. The digestive enzymes produced by these acinar cells ultimately get secreted into the duodenum through the pancreatic duct. So the previous slide shows kind of a cartoonish representation of acinar cells and how they produce digestive enzymes that get secreted into a network of ducts that also empty into the small intestine. Again, as soon as chyme, food digested in the stomach, as soon as that enters the small intestine, that chyme mixes with bile from the liver and bladder, and simultaneously it mixes with digestive enzymes produced in the pancreas. So what we see here, there are 4 main digestive enzymes produced in the pancreas, secreted into the duodenum, beginning of the small intestine. Two of these are potent protein digesters. One digests carbohydrates, one digests fats. So we saw how pepsin digests proteins. Well, trypsin and chymotrypsin, right, are the other potent protein digestive enzymes. These are also secreted as inactive enzymes, aka zymogens. A zymogen is an inactive protein. So trypsinogen, its inactive form, gets secreted. It enters the lumen of the small intestine and gets converted into active trypsin, which completes the process of protein digestion. Trypsin and chymotrypsin, right, secreted by the pancreas into the small intestine, are the major digesters of protein. Hydrochloric acid and pepsin start the process. Trypsin and chymotrypsin finish the process. Not much carbohydrate digestion has occurred by the time chyme enters the small intestine. Amylase, pancreatic amylase from the pancreas, is the major digester of carbohydrates. And then lastly, fat digestion. A little bit occurs in the stomach thanks to gastric lipase. Fat further gets broken down thanks to bile. Bile emulsifies fats so that pancreatic lipase do the last bit, most of the remaining fat digestion. So by the time that chyme in our small intestine mixes with bile and these four pancreatic secretions, this is what causes the complete chemical digestion of those nutrients, so that as chyme passes further down the small intestine, those nutrients can get absorbed into the bloodstream, travel to the liver will process those nutrients, then they can enter the vena cava, which brings it to the heart, and the heart will now pump all of these nutrients out to the body so that the brain, muscles, all of other tissues in our body can use those nutrients. Carbohydrates and fats for fuel, the proteins for making more cells, right, making more enzymes, etc. So the last slide I want to talk about before I can kind of open the floor to more questions is this slide here, which is slide number 44. This slide does a pretty nice job really summarizing the key digestive products associated with digestion, where they're produced, what they digest, where that occurs. So again, it comes down to carbohydrates, lipids, aka fats, and proteins. Now, what I really want to emphasize is there are a couple items missing from this image. First of all, what's happening in the mouth? The production of saliva. Saliva contains amylase, which does a little teeny bit of carbohydrate digestion. But I also mentioned that our saliva also produces a lipase. So I want everyone to include in your notes right here, excuse me, the fact that salivary lipase, produced in the mouth by the salivary glands, also does a little bit of fat digestion. A more important omission in this slide is shown here. In addition to pepsin, what other digestive product is produced in the stomach? Hydrochloric acid. Make sure that you include hydrochloric acid here as well. Hydrochloric acid produced in the stomach helps break down proteins in the stomach. Next, as chyme enters the duodenum, the beginning of the small intestine, it mixes with bile, right, from the liver, again, produced in the liver, but stored in the gallbladder. So bile can come from the liver or the gallbladder, even though it's only made in the liver. So bile from the liver, as well as pancreatic lipase, breaks down fats. Amylase, produced by the pancreas, breaks down carbohydrates in the small intestine. And then lastly, trypsin and chymotrypsin produced by the pancreas digest proteins in the small intestine. Now, you can just cross out this carboxypeptidase. I chose not to cover that at all. Pay no attention to that. Lastly, and this is described more in an upcoming slide. There's another class of digestive enzymes that are found within the epithelial lining of the small intestine. Those cells that have those microvilli, the cells that line the villi and then have the microvilli, as nutrients get absorbed from the lumen of the small intestine through the epithelial lining into blood vessels in the submucosa, there are enzymes within those epithelial cells. These are called brush border enzymes, and they drive what's called contact digestion. Well, they're called brush border enzymes because the microvilli on those cells make them look like little brushes. My point is just this. In terms of carbohydrate digestion, amylase in saliva, more effectively, amylase from the pancreas digests most of our carbohydrates. Starch, for example. Starch is a really big molecule, many linked together. Amylase will break apart starches and glycogen into individual sugar molecules. Well, an individual sugar molecule is called a monosaccharide. Two sugars together are called disaccharides, and many sugars linked together form a polysaccharide. Right? Starch, glycogen, these are types of polysaccharides. Amylase can break down a polysaccharide into monosaccharides, but a lot of the time it just breaks them down into disaccharides. Enzymes within our epithelial cells drive the last bit of chemical breakdown of disaccharides into monosaccharides. That's why they're sometimes called disaccharide-ases. Now, sure many of you know this already, but sucrose, right? Common table sugar, right? The sugar you see in sugar packets, the sugar that you put in your coffee. Sucrose is a disaccharide. One molecule of sucrose is actually one glucose group attached to a fructose group. If that gets absorbed through your intestinal wall, enzymes in the epithelium of that wall will break that apart, separate the glucose from the fructose. That's an example of these brush border enzymes. Now, let me see here. Here we go. My slide number 54 is talking about these brush border enzymes in more detail. Within these epithelial cells that line our villi, these cells that have the microvilli, there are enzymes within these cells that do just a little bit additional digestion, primarily of disaccharides like sucrose, breaking it down into glucose and fructose molecules. And that, in essence, is digestive system physiology. Know what digestive products, enzymes, hydrochloric acid, and bile, know where they're produced, know what substances they digest, and know where in the digestive tract they are digesting the molecules to digest. That, understand how blood containing these newly digested and absorbed nutrients flows into the liver, flows through the sinusoids and the liver lobules, how the hepatocytes that line those sinusoids enzymatically process nutrients and any other waste products, foreign substances that might be found in that blood. So, any questions about that information that I just shared?
This is Liz. I do have a question.
Go ahead, Liz.
Can we go back to slide 44?
Yes, slide 44.
Yeah, this one. Can you mention again? You said that there are some things that are missing here,
Yes,
and I didn't catch that.
sure. So can you see my cursor moving?
Yes.
So right here, so salivary glands produce salivary amylase. They also produce salivary lipase,
Okay.
breaks down lipids.
Fats. Uh-huh.
Next, our stomach produces more than just pepsin, right? Make sure that you include hydrochloric acid here. Hydrochloric
Oh,
acid
yes.
produced in the stomach aids with protein digestion. And then you can cross out or ignore this reference to carboxypeptidase. So it was really, yeah, just adding salivary lipase and hydrochloric acid.
Okay.
Otherwise, with those added, this is a really nice summary of the main digestive products associated with digestion,
Yeah.
the products they digest, and where that digestion occurs.
Cool. Okay. Thanks.
You're welcome. Okay. Does anybody else have any digestive system physiology questions? Okay. So let's see.
I do have a question.
Yeah.
This is Anna.
Hi, Anna.
I just turned on my, hopefully you can see it now. Okay, so I'm just a little confused. Maybe you explain this on how nutrients actually get like into the blood. So
Yes.
lumen, I guess.
Excellent question. So what it comes down to is this. We know that all that nutrient absorption is occurring in the small intestine. Well, oftentimes that absorption can be caused by just diffusion, right? Molecules diffuse from high concentration areas to low concentration areas. Most of that nutrient absorption, however, does involve specialized receptors on the cells that line the small intestine. On the microvilli themselves are receptors for glucose, amino acids, and triglycerides. Those molecules have to first get digested to the point that they're small enough that they can bind to those receptors. And those receptors essentially act like tunnels or like a gate. So once, say, a glucose molecule gets digested in the small intestine, it'll bind to a glucose receptor. That will then allow that glucose to enter that epithelial cell and diffuse through the epithelial cell into the submucosa, into the blood vessels in the submucosa.
Okay. So do you want us to know any of those receptors?
No, I
Okay.
don't know.
Just kind of the general idea.
Yeah, exactly. The general idea. Notice that, yeah, nowhere in my PowerPoint did I mention that because I just think it's a little bit above beyond the scope of this class.
Yeah.
And there's already enough material to cover. So, yeah, great question. Now, since you asked that, one thing to add to that, and this came up in my earlier class. For example, if you get food poisoning, if there are toxins from bacteria also present in that chyme, it can lead to vomiting or diarrhea, right? Lovely as it is to talk about diarrhea, it's important to talk about it. One of the common effects of food poisoning is diarrhea. Oftentimes, if typically viruses, but also toxic bacteria, if they are found in the small intestine, if they're consumed and stomach acids don't neutralize them, if they get into the small intestine, some of those toxins can bind to those same receptors, and it'll prevent those receptors from binding and absorbing anything. The muscles that line the small intestine will contract vigorously, and the body will expel all that material instead of absorbing it, essentially preventing those toxins from getting into the bloodstream and causing more damage. So another role of those receptors, they help ensure that only the good things like sugars, triglycerides and amino acids get in. And anything foreign, especially anything toxic, if those receptors recognize something as toxic, it'll induce vomiting or diarrhea, expelling that material before it can get into the bloodstream.
So that's how someone could end up with like malnutrition and malabsorption then too.
Yes. So if somebody is sick enough to the point where they're constantly vomiting or constantly have diarrhea, they'll get incredibly dehydrated because in addition to absorbing these digested nutrients, there's a tremendous amount of water that gets absorbed as well. So yeah, if anybody has food poisoning or along with this, COVID-19 viral infection, that severe diarrhea can cause severe dehydration, but also severe malnutrition. They'll probably have to get hooked up to an IV that contains electrolytes and sugars because their intestines can't absorb them. Lastly, anything that causes inflammation of the intestines, whether it's an autoimmune disease like Crohn's disease or ulcerative colitis or irritable bowel syndrome, severe inflammation of the intestines will also prevent proper nutrient absorption and can lead to malabsorption and malnutrition as well.
Thank you.
You're welcome. Okay. Does anybody else have questions?
Hi, Ryan. It's Rolando. I have another question.
Okay.
Are you going to go over in more detail on what role the central nervous system plays in digestion, or just what you had that one slide about the vagus nerve and that's about it?
Slide. The only role that the central nervous system has on digestion is digestion, specifically stomach activity. We can kind of kickstart our stomach, just the thought of food, the smell of food, the anticipation of food, right? Again, I can consciously make my elbow flex anytime I want. I can't just, try really hard and my stomach start contracting on its own, but hunger, the smell of food, the thought of food, that area, primarily the brainstem, can send a signal to the stomach, primarily kickstarting it, which is why our stomach growls sometimes when it's empty and we're hungry. But for the most part, almost all digestion, especially in the stomach, in the intestines, it's all occurring autonomically, involuntarily, with no direct connection to the brain, to the central nervous system. So if someone severed their vagus nerves, they would have a really hard time regulating their blood pressure and heart rate, but digestion wouldn't be affected at all. Well, it wouldn't be affected very much because most of the neurons, the stretch receptors that control stomach mixing and the contraction of the smooth of the intestines, most of those neurons don't even travel to and through the central nervous system.
Okay.
Short answer is there's really not much of a direct central nervous system connection to digestion. Most of the neurons that control digestion are just found exclusively in the digestive tract. They don't connect to the central nervous system.
Thank you.
You're welcome. So Brittany asked a question through the chat feature. Yeah, so what happens if someone has their gallbladder removed? Well, what it comes down to is this. Most people can lead a perfectly normal life if they've had their gallbladder removed, really with one exception. Because your gallbladder stores and concentrates bile, if that bile is concentrated in the gallbladder, it means that if you eat a meal really high in fat, the gallbladder will secrete this highly concentrated bile into the small intestine to aid with the breakdown of that fat. Given that bile is made in the liver, if you have your gallbladder removed, your liver is still going to produce and secrete bile into the small intestine. It's just more diluted. It's more weak. So the point is, if anyone's had their gallbladder removed, the only real effect that has is they probably have to watch their intake of fat. They have to probably cut back on how much fat they eat. Otherwise, if there's not enough bile to break down fat, then lipase enzymes aren't going to be able to fully break it down. Not as much fat is going to get absorbed into your bloodstream, right? Which most people think, oh, wouldn't that be great? Well, no, our body really needs that fat for a lot of reasons. Too much can make us obese, but not enough can kill us. Also, if our enzymes can't break down fat properly and it can't get absorbed, it means that some of the bacteria in our gut can. So if you don't have a gallbladder, you won't digest fat enough, you won't absorb enough fat into your bloodstream, but it can also provide more undigested fat for the bacteria in our gut. And when they digest fat, it produces a lot of gas. So it can lead to bloating, discomfort. But for the most part, if you have your gallbladder removed, you pretty much just have to, watch how much fat you eat. You probably shouldn't be eating a lot of, potato chips and ice cream. But aside from that, if you just eat a moderate diet, it should have no dramatic effect whatsoever. So yeah, thank you for asking that question, Brittany. Okay, so any remaining questions? Primarily physiology questions. We'll really review anatomy and histology. On Wednesday, we can revisit any and all respiratory system topics more on Wednesday. And again, I'll go over exam format then as well. Kind of sticking digestive enzymes and liver function. Does anybody else have any questions from the study guide, from review sheet, from Wendy's video? Okay, so I also see in the chat, so just to share with us, who is Office 1? Thank you for commenting on your personal experience with having your gallbladder removed. Oh, Brittany. Thank you, Brittany. Oh, okay. So I'm kind of talking as I type here. Yeah, so Brittany, do you care to chime in on that? Since you've had your gallbladder removed, do you have any specific digestive issues, anything significant? Can you confirm that consumption of dietary fats is something that you have to pay attention to, or does it have any effect at all? Maybe she's typing up an answer. I'm guessing Brittany's at work and can't probably talk through the WebEx meeting. But yeah, I'm just I am thankful that you were willing to share that. Okay, so that being said, since I'm not getting any more questions, I'll just give a last call for questions.
This is Liz.
Okay, go ahead, Liz.
I don't have any questions right now. However, I mean, I'm not in this session normally, and I was wondering if anybody could share this video with me when you post it, because I'm not going to be able to see it.
Oh, okay. Well, what you're going to see is the video that you will have access to. The video from my morning WebEx meeting is, I mean, virtually identical. I pretty much covered the exact same material. But Well, actually, Liz, you know what I can do is send me an email, and I should be able to email you the link to this afternoon session
Oh, okay.
so that you could see it and the recording from the morning session.
Okay. Thanks.
Okay, so yeah, Brittany, I had a typing out an answer. Okay, drinking, having your gallbladder removed. So I'm guessing that's interesting. I wouldn't have thought of that connection. I'm guessing that you probably just obviously anybody drinks too much, you suffer from a hangover. I'm guessing that it just you don't have to consume much alcohol to have that same horrific feeling of a hangover. Um, just curious, but yeah, I imagine that yeah, having your gallbladder removed because obviously the liver detoxifies alcohol. Typically, not having a gallbladder typically doesn't affect the rate and the degree at which the liver produces bile. Um, it just again prevents the body from storing and concentrating bile. So there's a limited amount, which more than oftentimes impacts fat digestion. Um, but interesting that yeah, having a gallbladder removed does, at least in Brittany's case, yeah, it does impact the ability to consume and metabolize alcohol. And yeah, and a last comment, yeah, I'm sure it is horrible. I can imagine, I know for me and just my own metabolism, I have a pretty fast metabolism. And for the most part, I've always been able to eat whatever I wanted. And I think I may have mentioned this in class before. It's a little bit off topic, but most of the males in my family are kind of tall and lean with really fast metabolisms. And most of the females, like my mom and my sister, have really slow metabolisms and they have to pretty aggressively watch what they eat and are constantly dieting and exercising where I can still eat ice cream and potato chips and not really um, doesn't really affect me much. So I can only imagine how horrible it would be to have to limit my intake of alcohol and fat. Those two make up well, fat more than alcohol, of course. But yeah, if I had to really cut back on fats and not, if I couldn't eat potato chips, for example, that would be a major quality of life impact on me. So I can just only imagine how challenging that would be. But hopefully, Brittany, the rest of your digestive function is unaffected by the lack of a gallbladder. Hopefully, you can still process and digest proteins and carbohydrates, but it sounds like alcohol and fats are the issue. So again, thank you for sharing that. Okay, so that being said, feel free to jump in with any last minute questions. If I don't get another question in the next minute or two, we'll go ahead and terminate this meeting. Okay, so that being said, just another reminder, Wednesday, the entire session is going to be review. So come to Wednesday's WebEx meeting prepared. Read through material, come with your questions, with your review questions. Okay, so that being said, if I don't get in any other questions, I'm going to go ahead and end the meeting. Again, I'll be posting this on WebEx. Sorry, I'll be posting the WebEx recording on D2L sometime tomorrow. So until then, everybody, have a great week. Have a great couple of days. Stay safe, and I will see you all Wednesday at 1 o'clock.
Thank you.
You're welcome. Signing off.
Thank you.